Hello and welcome back to 11.2 Notes. This is part 2. Last time in 11.2 Notes we covered the beginning portions of 11.2, including the skeletal system and the structure of the muscle cell, which you can see on the screen here. So now we're going to move forward. We're going to get a little bit closer in. We're going to zoom a little bit closer. So you can see in this diagram that I have a muscle fiber up here. So we said a muscle cell is made of all these muscle filaments, those cylindrical bundles you can see. And then you can see over here, if I take those out, this entire thing is a muscle fiber. If I take one of those cylindrical filaments, that is called a myofibril. Myo just means muscle, so it's a muscle fibril, so a smaller portion of a muscle fiber. And then within that, it's made up of these repeating sections. So I can see this section here, and then I have a Z line, and then I have an identical section here, and then another line, and that continues all the way down that myofibril. And this is the actual structural repeating units of a muscle cell. So that is what is called a sarcomere. A sarcomere is the repeating functional unit of a striated muscle cell. So a sarcomere is this section from one Z line to the next. That is a sarcomere, and it's made up of a couple of different segments, like you can see down here. The important thing to know is that these segments are made of two different structural proteins. Okay, so the first one is myosin. Myosin is the thicker filament. So I can see myosin in my diagram here, and I can see myosin in my diagram here. And then the other one is actin, and actin is these thinner filaments. So I have actin here in my diagram, and I have actin here in my diagram, and it kind of goes around the myosin. So you can see as well in this diagram, the myosin has these heads on it, these little offshoots that kind of grab onto the actin, and that's what actually causes the muscle to move. So it causes the myosin moves along the actin, and it shortens, and that's what causes a muscle to contract. So that happens in this sarcomere and this sarcomere and this sarcomere, etc., all the way down the line, and overall that entire muscle fiber contracts, and it happens on a larger scale than that as well, and the entire muscle contracts and it gets shorter, and that allows it to pull on the skeleton like we talked about last time. So the sarcomere, we see a couple of different things that are all made up of the interactions between this myosin and the actin. So I can see my myosin in the middle here, and it has the heads on it, these little offshoot branches, and then I can see my actin. And so my actin is the thinner filaments kind of go around the myosin. And so this down here is an electron microscope diagram of a muscle fiber, a striated muscle fiber. And the reason it's striated is because we see these different colors, okay? So these different stripes, and striations are just stripes. So I have a really thick dark line here. That corresponds to my Z line. And the Z line is the edge of the sarcomere. So a sarcomere comprises from one Z line all the way over to the other Z line. This whole segment in here, that is one sarcomere, and that is a repeating unit. And then inside of that, you can see I have a dark band here. So this section looks darker than the sections outside of it, and that is where I find my myosin. So the dark band is the myosin. Specifically, these really dark sections, those are the areas where my myosin is overlapping with my actin. So where those two both show up, I get these really dark bands. And so I can see where these proteins are with just an electron microscope image. Even though I can't see the individual proteins, I can see the shading that the proteins create. And so what happens is that actin moves along the myosin. So I can see the sarcomere here, and there's a little bit of space between the actin on this side and the actin on this side, and also there's a little bit of space between the myosin here and the Z line. But what happens is it contracts, and so when it contracts, I have these myosin heads on here, and they kind of grab onto the actin and pull. So this will grab on here and it'll pull, and so the myosin will move this way. And it will move closer to the Z line. And so when that happens throughout the entire sarcomere, I get this image down here. So now the actin on this side and the actin on this side are right next to each other. There's very little space, and the myosin here is very close to the Z line. So let's take a little bit of a closer look at this process. Okay, so we have a couple things going on here. We have ATP hydrolysis and we have cross-bridge formation. And those two processes are what are necessary to make the filaments slide across each other. Okay, so in order to make that happen, I need calcium ions. So calcium ions have come up before in the nervous system and muscle contraction. They come up again. They're really important. It's really important to have enough calcium, or else none of these processes work. So first thing that happens is I have an action potential. Okay, so hopefully you'll remember action potentials from back in topic 6. And so an action potential is a signal that gets sent from the brain, and the purpose of this one is to make a muscle contract. So the action potential reaches a striated muscle cell, and the sarcoplasmic reticulum, which hopefully you'll remember from part 1, releases those calcium ions that it holds into the myofibrils. And what that calcium does is it opens up these binding sites. So all along actin, there are binding sites for the myosin heads to attach to. And normally they are closed off, but calcium is able to open up those binding sites. So those binding sites open up, and now I can have this myosin head bind onto the actin. So now the two are connected. The myosin is connected to the actin, and that is what is called a cross bridge. Okay, so this is a cross bridge. It's when the myosin binds to the actin. And when that happens, the myosin head will pull along and move the whole myosin towards the Z line. So it'll pull along the actin, it'll pull this way, and the myosin will move that way. So once that has happened, they're kind of locked in that position, and so you need something to reset it. So that's when I have ATP comes and binds to the myosin head, and it resets it. So it moves it from attached to the actin, and it takes it and it detaches it from the actin, and it resets it back up in its configuration where it can bind. And so once that has happened, then the myosin head is free to attach to a new spot on the actin, and it continues kind of crawling along the actin and forcing that myosin to go closer to the Z line. And so this happens on both ends of the sarcomere. So it's happening over here. This myosin is moving towards that Z line, and this myosin is moving towards that Z line. Technically, the Z lines are moving towards each other because the myosin doesn't get any longer. But in any event, the result is that the sarcomere gets shorter. And so when the sarcomere gets shorter, that is a muscle contraction. So contracting means to get smaller. So it gets smaller. And when the muscle gets smaller, it pulls on the bones that it's connected to, and it's able to affect a movement. Okay, so that is the mechanism by which the myosin crawls along the actin to pull the two Z lines closer to each other. And in order to do that, it needs calcium ions and it needs ATP hydrolysis. And those work together to get this process. So you could be asked on an exam to explain how skeletal muscle contracts. So why don't you give an attempt at answering the question now, pause the video, and see if you could write out an 8-mark response to a IB question that is explain how the skeletal muscle contracts. And here would be a sample mark scheme to a question such as that. So we would have that the muscles contain repeating units called sarcomeres. That's 1 mark, which you can see indicated by the semicolon at the end there. Muscles and sarcomeres contain actin and myosin filaments. The actin fibers are thin and the myosin fibers are thick. An action potential arrives and causes release of calcium ions. That calcium is released from sarcoplasmic reticulum. That binds to troponin, which is a little bit more detail than we went into on the last slide. That causes troponin and tropomyosin to move on the actin. Exposes binding sites on the actin. ATP binds to the myosin heads, which releases them, breaks the cross bridges. Hydrolyzes ATP, causes them to change shape. The myosin heads bind, form cross bridges, and swivel or move the actin, and they move it towards the center of the sarcomere, and that shortens the sarcomere. So there's obviously more than 8 marks there. There's a couple different ways to get to the 8 marks. So again, here are some electron micrographs. So you need to be able to look at images like the ones we have here on the left and be able to infer from those images what the state of contraction of the muscle fibers is. Okay, so as we can see here, the H bands remain constant. So it goes from here to here. And so if you remember back from that earlier slide, what the H bands are is the same as the kind of dark bands. That is where the myosin is. Okay, so the myosin is controlled within the H bands. And we can see the same thing on our diagram over here. We can see that the myosin maintains the same width. But what changes is the Z bands go from being out here, and over time they get closer together. Okay, so the contraction is really a function of the Z lines moving close together, and again we can see that in our diagram over here. The Z lines move closer together. The myosin pulls the actin in towards the center, and that condenses those Z lines closer together. So you should be able to look at an image like this one on the left and be able to infer whether the muscle is relaxed, contracted, or somewhere in between. So for example, we have here a micrograph. You could be asked to deduce whether the myofibrils here are contracted or relaxed. You could be asked to calculate the magnification of the electron micrograph, which was way back in topic 1.1. You could also be asked to measure an individual sarcomere, although that does tend to be difficult, and so you kind of measure the total length of the sarcomeres because it's easier to measure, and then divide by the number of sarcomeres, and that way you can get the average sarcomere length. So I know it's really hard to do these things in videos, but it may be a good idea for you to stop, pause the video, and see how many of those questions you can answer right now. You should definitely be able to answer at least the first one and take a shot at those other ones. So we have some nature of science here to discuss. We have developments in scientific research following improvements in apparatus. Okay, so what that means is oftentimes we are able to gain more scientific knowledge when we are able to improve the equipment that we are working with. And that sounds pretty obvious, but it's also important to remember that our scientific understanding is limited by the equipment that we have at hand. So for instance, fluorescent calcium ions have been used to study the cyclic interactions in muscle contraction. So this is not something that could have been possible to study before we had the ability to create and visualize fluorescent calcium ions. So Ashley and Ridgway in 1968 were the first ones to be able to study this. Before that, we just did not have the equipment. And so what they do is there is a calcium binding bioluminescent protein. So what that means is it's a protein that creates light. It is luminescent. And upon calcium binding to that, it emits light. And the timing of the light emission peaks between the arrival of the electrical impulse at the muscle fiber and the contraction of the muscle fiber. So this shows us that basically the theory that we have in place is what is actually happening. Because remember, we can't zoom in and visualize these things we've looked at diagrams of. We don't have the equipment necessary to view things that small. So we need things like this that will kind of indirectly give us evidence to whether or not our theories are consistent. And those light emissions can be recorded with special microscopes and cameras, and we can do some things to visualize that. Even though we can't see calcium ions themselves, we are able to visualize their presence using this process. And this process in and of itself was not possible until we had the appropriate advancements in apparatus. So technology and scientific understanding are really closely intertwined. As technology improves, we are able to get more information out of the world around us. So that concludes topic 11.2. I hope you found this helpful. If you have any questions, feel free to leave them in the comments, and I'll see you next time.